When we started thinking about this plant biology initiative symposium, whenever it was, and I guess it was a few years ago, knowing 150 was coming up, I can tell you the first person who popped into my mind was Susanne Renner, because I've been reading all of this literature around senescence and leaf coloration, and there's just so many things where her work intersects with the kinds of things that we do, like Michael, and so many others we've heard about in the last 2 days. But just to tell you, Susanne Renner was the director of the Munich Botanic Gardens for many years, and put that set of woody plants to good use and work. So even if it's not our trees, it's very much the same thing, the importance of having an arboretum within that garden, which is a magnificent botanical garden. And to say the least, I won't even try to encapsulate all the things that Susanne has written about and interrogated in the botanical and natural world. But today she's going to talk to us about tree growth, autumn leaf senescence, and global uptake of carbon. So it's like I was just waiting for leaf senescence, and I'm getting 2 bonuses.
Thank you, Ned. Yeah, it's a real pleasure to talk about these topics here based on work in the Munich Botanical Garden, but also other gardens. I want to start with a little video clip. It's less than a minute.
Hi, this is Bill Putnam. I'm a climate scientist at NASA's Goddard Space Flight Center. What you're looking at is a supercomputer model of carbon dioxide levels in the Earth's atmosphere. The visualization compresses 1 year of data into a few minutes.
Look at the dates.
Carbon dioxide is the most important greenhouse gas affected by human activity. About half of the carbon dioxide emitted from fossil fuel combustion remains in the atmosphere, while the other half is absorbed by natural land and ocean reservoirs.
Look, it's May, June.
In the northern hemisphere, we see the highest concentrations are focused around major emission sources over North America, Europe, and Asia. Notice how the gas doesn't stay in one place. The dispersion of carbon dioxide is controlled by the large-scale weather patterns within the global circulation. During spring and summer in the northern hemisphere, plants absorb a substantial amount of carbon dioxide through photosynthesis, thus removing some of the gas from the atmosphere. We see this change in the model as the red and purple colors start to fade.
That's what our trees do for us. Okay, stop now, but I'm gonna click it again. I don't know why it stopped. It's just a moment. Actually, my computer froze right now. It's gonna get all green. So by June, it's all green. It's really wonderful, but I have to wait until I do not know what this computer froze. There's nothing I can do now. I guess I have to restart. Yeah, I guess I have to restart.
It's the spinning beach ball.
I think I have to restart. Don't worry, the talk is not endlessly long. Even if I restart now, which I have to do, it's not going to be over time at all.
No, don't worry.
It's 36 slides, so it is actually, it's said that your computer shut down due to a problem. Now I see the apple and it's starting again. We will skip the movie in the second go. Anyway, had this movie continued for a little more until June, August, it would have been completely green, because the vegetation in the northern hemisphere would have sequestered that carbon dioxide. And then actually, as it goes on, it's less than a minute. As it goes on, it shows the fires in the southern hemisphere and the emission of carbon monoxide from these fires. So it's super interesting. This computer is still starting up. So the reason I started with this is obviously to provide context of why understanding what trees do, and many species of trees throughout the northern hemisphere, why this is so important. It's important because we need to draw down some of the excess gigatons of carbon that have accumulated in the atmosphere since the Industrial Revolution. We now have about almost 300 gigatons carbon more than we had 1850 to 1900, with most of it, of course, having been emitted just in the last 20, 30 years. And my second slide, we will skip this movie, shows that. It's still starting up, but... I don't want to just keep talking because then the whole thing is. I'm sure it's gonna start up now. It's just halfway here, and then. Okay. All right. Password. Okay. Okay, ignore, cancel. All right. Okay, here's the talk. Arnold Arboretum. Admire my orderly desktop.
You can tell this is not my computer.
Okay, it's starting up, it's opening up, and it should work just fine now. Okay, opening. All right, full screen mode. Okay. All right, and we skipped the movie. All right. So this was the background for work on plant phenology on trees in the Botanical Garden Munich. The backdrop is the rate at which we have been accumulating, we being humans, CO2, throwing these gigatons of carbon into the atmosphere. And the atmosphere that we have to live in, that all life has to live in, is very thin, proportionally as thin as the skin of a balloon. And as you know, planting trees or having vegetation is one of the natural climate solutions. It is still the cheapest way to draw down the extra carbon. And we need to draw it down because carbon dioxide is really warming the atmosphere, more so than other bad gases like methane. So what is the readily achievable potential for increasing CO2 sequestration and storage? This is a super important question, and this is what our work addresses. This is the big question. Now here's an aerial shot of the Munich Botanical Garden. It is not that large. It is quite a bit further, located quite a bit further north than Boston, and it has about 1,400 woody species permanently outdoors, shrubs and trees, 85% of which are not native to Central Europe. And this gave us this great opportunity to consider our botanical garden a common garden experiment. And since I was the director for 17 years until just over a year ago, I gave myself permission to cut off parts and bag parts of trees and so on. All right. The work that I'm going to present today is mostly work of the past 2 years, and it is work that has been done mostly by these people. Deborah Zani, she was an Italian Master of Science student with Konstantin Zohner. Li Dong-mo, who is a doctoral student with Konstantin and the professor there, Tom Croter. Konstantin, a former PhD student and now colleague and independent researcher at the ETH in Zurich. And as I just mentioned, Tom Croter, who is responsible for many of the very large data sets we are able to use. And first I will talk about this paper here, on senescence, new insights about the drivers of senescence. Then I will talk about an experiment on how spring and autumn phenology translate into growth. And then we build on the results of this experiment to produce this new work, which will be the end of my talk very briefly. And then I will end where I began, namely with the carbon storage potential. Okay, so here is a small part of the Munich Botanical Garden with some of the ongoing, or at that time in 2017, ongoing experiments. We did experiments, we being mostly Constantine and master students. The Master of Science is obligatory in Germany. So the experiments concerned 400 species of trees, but the work I'm presenting in this talk will be focusing mostly on the saplings of some few species, 9 species, that we also worked on. And a key aspect in my talk today are these styrofoam plates that you see there. And I never forget the day in May 2017 when I was giving a tour to the right-hand man of the Bavarian Minister of Science. It was just him and me, and I gave this tour, and then we came to this patch here, and he said to me, "Are you doing these experiments yourself?" And I think he wanted to give me a way out. Like I could say, "Oh no, it's the gardeners," or something, because it looked so messy. Okay. All right. So one of the experiments, not the one that I just showed, concerned the question, what will happen if we damage the leaves of saplings by frost? It's an important thing. With climate warming, things are leafing out earlier, then there comes this occasional night frost, and the young leaves are, of course, the most sensitive, the most vulnerable part of the tree at that time and can be killed by frost. So we took saplings of these 3 species here and killed their leaves in a fridge. Okay, and then it takes these species, and I'm going to just focus on one of them here in this graph, but we did it with 3 species. So you have in blue the experiments where you kill the young leaves, and then it takes this species about 4 weeks to make new leaves. And the question was, what happens with that second crop of leaves that comes out much later than normal, like in June, okay? It takes 4 weeks, 36 days to 42 days for these things to make new leaves. All the leaves are killed on that sapling. What happens is that they overcompensate. So here's that second crop of leaf, and they have a higher rate of photosynthesis, and they stay longer, and they outdo the control. Here in the dashed line is the control. Saplings, same species, same place, but leaves not killed. Okay, so the second crop of leaf manages to outdo the normal leaves. And then if we give them experimental warming, as we did here, 4 degrees above normal the control air temperature, they drop their leaves earlier than control. You warm them, they drop the leaves earlier. You kill their leaves, they keep them longer and have a higher photosynthesis rate. So this raises lots of questions. If it is, what determines senescence? If it is determined by frost or by lack of water or microbes or environmental conditions, how can it be that these things actually drop their leaves later and the warmed ones drop their leaves earlier? It was clearly what happened here in the first part of the year when we did these experimental modifications that determined the leaf fall. It raises lots of questions. All right? So what do people think about what determines leaf fall? And I'm very happy to have met Pam Templer here, who with her students did a meta-analysis in 2015 of 68 studies that had been published at that time, only a few of which included, 18 of which included data on change over time, change in senescence over time. And 6 of these reported the effects of air temperature on senescence. And in all 6 cases, warmer temperatures in the fall led to later senescence, which you would kind of think initially. Nice warm fall, the leaves are still there, provided you have water. So what do people think? And that's all in this paper except these last 2 points. So shorter day length, lower temperature, first frost. That should do it. That should determine senescence. It's the default expectation. Or, in addition, perhaps, stress, drought, nutrient scarcity, pathogens. This could, of course, interact. Or strong radiation when the chlorophyll is already being dismantled. And as those of you who are botanists will know, this dismantling of the chlorophyll that starts basically in August. It takes a long time. The tree wants to get its nutrients back from the leaves it's about to shed. And the taking apart of this photosynthetic machinery is really a difficult and complex process. And at the end, there's a stage when there is this reactive oxygen species. Is basically photons that can damage the DNA or do damage the DNA. So it's a vulnerable stage. This last stage in the leaf's life is vulnerable. And then there's the idea that plants might throw away their leaves when they don't need them anymore. This is a new idea. It's not in your meta-analysis. Okay, so let me explain this idea a little bit more. Actually, it's also not a new idea. This is known as sink regulation or limitation of photosynthesis. And it's an old idea. It is in this textbook from 1868. And it has also been pushed by Christian Kerner for the past 20 years. Sink limitation. Okay, what does it mean? It means that the tree puts out its leaves, and then it produces the assimilates and does whatever it has to do. It grows, its roots grow, its little twigs grow, or it has to put out the flowers. It starts maturing the ovaries into fruits and produces exudates for the mutualists. I mean, it does lots of things, okay? And when it's done with those processes, with those growth processes, it throws away its leaves. Now, this sets up two very clear hypotheses. And the one is, one is the null hypothesis that leaf senescence is determined by day length, temperature, and other environmental factors. That is clearly the null expectation. And the alternative hypothesis is that leaf senescence is determined by sink limitation, as I just described, regardless of autumn environmental factors. And the way why we are taking this serious is these experiments with these killed leaves, where clearly the external conditions, temperature and light, had nothing to do with when those plants were dropping that second crop of leaves. So there's got to be something to this sink limitation hypothesis. Let's test it. But testing it is difficult. Okay? We need experiments and real-world data to check which of these two hypotheses better predicts senescence. And here is my description of this experiment. Okay, it involves exactly those styrofoam things that styrofoam plates that I showed before. Because with this simple experiment, you can either shade an entire plant, or the control is out in the sun, by placing these things depending where the sun rises in the east, etc. Or you can place them carefully over a seedling. These were 5-year-old seedlings of 2 species. You can place them carefully such that half the plant is in the shade and half the plant is in the sun. And of course, you really measure the effect, okay? You measure the photosynthesis rate, all right? And why is this so brilliant? Because this gets at the question, is plant growth determining when the leaves are being shed or will be shed? Or is it the amount of photosynthesis that determines when the leaves can be shed? See, when you shade the entire plant, its growth will be less, presumably because it receives less sun. If it's fully in the sun, it does whatever it has to do, or it's able to do. They're all watered. They're all in the same patch of soil. When it's half shaded, clearly, the leaf level, the amount of synthesis done by these leaves will be different, because half of them are in the shade. Okay, so you get at this question, is it the plant, the trunk, the roots, whatever, the plant that determines when to throw away the leaves, or is it the leaf level photosynthesis? So I almost want to stop. What is your expectation? All right? It is absolutely the sink that determines when to throw away the leaves. Here you see the effect. When you have full sun and full shade, okay, then you see here is the, I'm at a very awful angle. I should have taken the microphone. Okay, here you see the effect. Here is photosynthesis change percentage, and here is senescence change, okay? So when they are in full shade, the senescence in these plants was delayed by 13, here you see 13, plus minus 7 days in the beech, and 8 plus minus 5 days in spirea. Bad conditions, they keep the leaves longer. Makes sense. In the half-shaded plants, which clearly had 50% of their leaves doing less photosynthesis, there was no difference. All right? So clearly, the leaves are shed when the plant level growth is done. It's the plant sink that determines when the leaves are shed. Now, we also did experimental spring warming and CO2 additions with these, not with the styrofoam. Obviously, this has to be in an enclosed growth chamber, and this part was done in Zurich in growth chambers. Okay, and so when you have these great conditions with the CO2 fertilization in the spring and the warming, experimental warming, then senescence in these two species occurs 3 weeks earlier. They have these great conditions, they do whatever they have to do faster and better, and they throw away their leaves 3 weeks earlier. Okay? Grows faster, throw away leaves earlier. Now, we want to test whether these new ideas better explain, okay, which hypotheses, the same two hypotheses, and we are now in favor of H1, at least our experiments are in favor of H1, which of these two hypotheses better predicts leaf senescence in the real world? And to do this, we used data from the pan-European Phenology Gardens, okay? And we used data for 6 species, some 14,000 individual time series. Time series is basically one tree. If the tree dies, they choose a tree at the same locations. It's all georeferenced. Okay, the data are from 1948 to 2015. Mean duration of these time series is 30 years, 700,000 leaf-out dates, and almost 500,000 senescence dates. Now I want to put in one slide about these European Phenology Gardens, because we are at a symposium focusing on what can be done in botanical gardens in this subfield phenology. So Europe actually has 2 such databases. One is the Pan-European Phenological Database, and it has data from 1948 to ongoing on 265 species, including cultivars. The other one is the International Phenological Gardens Network, which started in 1959, is still ongoing. It has 131 gardens. I mean, gardens is not the right word. These are little plots usually associated with a botanical garden, within a botanical garden, next to a botanical garden. Okay? It's not a garden. It's a little piece of planted area. Okay? And it includes the data also freely accessible. Includes 23 species, 12 of them also included in this database. But the crazy thing, and why I wrote this little paper here, is the crazy thing is that this International Phenological Garden Networks is based on clones. It is just amazing. It was set up by foresters, and they thought, oh, German foresters, we're going to be really precise. And to be precise, we're going to create clones. They have clones of pinus, I mean, incredible work. Clones of Forsythia is more easy. Incredible amount of work. And when you start a new garden, which is still happening, then this guy, very nice person, sends these clones, which were produced in Berlin and Hamburg. He actually packs them and sends them to the new botanical garden so they can plant these clones. Very few papers based on this, there are not that many papers, 50 papers or so, have realized the statistical problem this introduces. Okay, you're dealing with clones, but also there is actually super potential for science if one realizes that you are dealing with these clones. Okay, and that 12 species, this one does not use clones. This one is all clones planted at different places and observed over this time frame. I mean, it really opens research possibilities, and I'm stressing this so much because I think it will end, because the clones are currently dying. And when Dr. Chmielewski retires, which is next year, this will end. Nobody is going to send these clone things around anymore and maintain them. All right. So back to the data we used, which were from the Pan-European Phenological Database on these species, as I already mentioned, at these locations, observations, leaf-out date, and how do we define, and how do people define senescence? And Richard Primack yesterday already explained this. For some species, you use 50% color change. The observer, it's usually paid observers in this network here, and they have experience, and they will sort of eye the tree and do it in the same way, presumably, but it really doesn't matter. Given the number of observations, it doesn't matter if it's a little bit off. And for some species that do not change color, like this beech doesn't change color, it is the 50% leaves dropped. All right, these are the data. Now, how do climate models actually predict leaf senescence? And there are two types of models at this time, developed in 2009 and 2019 by Nicolas Delpierre and his collaborators. And the first generation of these models that everybody uses, no, everybody doesn't use the first generation model, but the first generation of the models that people use included as the parameters thought to predict senescence or tested, or how it fits with the observed senescence, number of frost days, okay, cold days. And then they put in the autumn day length. And then in the second generation model, they also put in spring leaf out, growing season temperature, and water stress. Okay, these are all environmental factors as expected under the null hypothesis, that is really the environment that determines leaf fall. But to test our alternative hypothesis, we need to put in some parameter that captures the sink filling, and it's difficult. Okay, but what we did was we put in this growing season index, and it's really the day length over the entire season from when the leaf actually comes out, which is not the same for every species, obviously, to when it drops, or when 50% drop. And we also kept all this other stuff. Okay, so we just added this parameter to the other components of the second generation model. And we also played around with water. Okay, so let's see. First, I want to stress that this is really important work, particularly for New England. Because last year, my husband and I, we drove from St. Louis to Niagara Falls. It was great. And then upstate New York and over to Vermont. And I had all planned this to see the fall foliage. I finally wanted to see it. I've never seen it. Okay? And we came, and the best I saw in 2 days of driving was this stupid tree here. And we were off by, of course, I looked in the newspaper afterwards. We were off by 8 or 9 days. So the current models are terrible. Okay, here we have the comparison. Here we have the first generation models, okay, the cold days, basically, and also fall, shorter days in the fall. Here we have the second generation model that already put in spring leaf out and temperature and water a little bit. And here we have our new model, which we call the growing season influenced model because of this one addition, and also we played around with water. And here you see how well these models explain the observed senescence in these half a million senescence states over that time in these 6 species. And the first generation model explained the data pretty badly. Second generation models are better, and ours is even better. Here's the R. Okay, so clearly it improved the predictive power. All right, and so we can tease this apart a little more. What are the things that really influence senescence, that really predict it? So we have the photosynthesis. Photosynthesis has the biggest influence, and this is kind of a proxy for what the plant accumulates there over the season. This has the biggest influence, and the opposing effect, second biggest influence, is autumn temperature. This is advancing leaf fall, and this is delaying leaf fall. Okay? And in between is summer temperature, little effect, and water has very little effect. Spring leaf out, a little effect. Okay. So basically we can say that in these years, the fact is that with high spring-summer productivity, individual trees that were observed were associated, or these conditions in individual trees, which the actual empirical data are associated with earlier senescence. Okay, climate warming, earlier senescence. Now, what are the implications of the insight that the sink, the growth, and its associated things like exudates and so on, determines leaf fall? But first, there should be with the ever earlier springs and higher productivity due to high radiation and carbon dioxide levels, at some point we should have earlier leaf senescence than now, because we have these opposing effects. High productivity, earlier senescence. Warm fall, later senescence a little bit. Okay, so these two effects at some point should cancel, and then the higher productivity should override the effect of the fall warming. And also temperate trees will eventually capture less carbon dioxide than predicted in the current models. Now I want to stress that this is all based, this is actually sort of a paradigm shift. In the commentary they said this was a paradigm shift, it's not even from me. But this is all based on experiments on 2 species in the Munich Botanical Garden and this monitoring of the 6 European species. And clearly this experiment should be repeated with American trees, carefully chosen, different clade, different, everything different trees, conifer, and American or Chinese monitoring data, empirical data. Oh, I just forgot this fly in here. Who should finance this work is the New England Naturalist Park. Okay, for sure. So now it is very difficult to predict the future, but of course we tried this. So here's 2020. Here we project this into the future, as people have done before with these traditional models predicting senescence. We are not doing something outrageous here. So the first generation models, if we project them out into the future, they would have predicted that by 2180 years from now, or 78 years from now, the leaf senescence would occur 20 days later than now. The second generation models predicted that by that time, senescence should occur 7 days later than now. But our model predicts that by that time, senescence should be 6 days earlier than now. And of course, this correlates with the growing season length. Yeah? So traditional model says growing season becomes longer by a lot, and our model says growing season by 2100 is not that much longer than it is now. Okay? Here's 2020. So this turning point. This has tremendous implication for the predicted gigatons of carbon that can be pulled down by these trees, because the longer you have your leaves out, the more you can sequester carbon dioxide, as we saw in this movie that stopped. Anyway, now I want to take a little detour to talk about this aspect here, strong radiation when the chlorophyll is already being dismantled. It's a short detour of 6 slides. Okay, so the first paper is a more recent one, came out 2021 by a Chinese lab, and it addresses what happens in Europe with the reduced air pollution that we have there now. Okay, so efforts to reduce air pollution have been going on for a long time, for something like 60 years, but they really had a positive effect in 1982. Okay, the pollution in the air is reduced, and therefore you get higher radiation. And the statistical analysis shows that up to 90, this is a statistical effect to see where the turning point is, okay? You see that before 1982, on average, the autumn radiation in watt per square meter is this, and since then it's become higher. More radiation since 1982. It's coming down on European forests. And so this work, Konstantin is a co-author, looked at the same, or used the same data, these European garden data, to see whether these leaves were being shed early or later in response to this radiation. But of course it's hard to disentangle. In fact, they could not disentangle without experiments whether it's climate warming or higher radiation that has an effect. They tried, and I will show you how they tried it, very smart. So what they tried is, in this statistical attempt to disentangle these two things, they found that, first let me say, over the entire period of 66 years, senescence changed by 1 day in these species, by 1 day. Minute effect, okay? It's 1 day later now than it was 66 years ago. But in this statistical attempt to disentangle the effects of the two parameters, they found that high radiation advanced senescence in, I think, 71% of the individual trees that were studied. Here's the tree numbers. Okay, here's the trees, or the observations of senescence, and here are the sites. Okay, and in 60-some%, I can't see it. Okay, warm temperatures delayed senescence. High radiation advanced, warm temperature delayed. As we have been expecting by now, if you've been paying attention. Okay, so but they unfortunately only looked at, or could only with their data, only looked at high radiation 3 months prior to senescence and warm temperature 3 months prior to senescence. They never considered what happens early in the year. I will come back to that. All right, so continuing on this detour on radiation. In the Munich Botanical Garden, I organized a team of 3 bachelor students to go out and photograph leaf colors. It's so beautiful in the fall. And check, how the leaf colors correlated with the geographic origin of these trees. And this has, of course, been in the literature since 1880, that there are geographic differences in the percentage of reds, anthocyanin-producing species in North America, Europe, and Asia. This is not a new idea, but I thought it's a great bachelor project, let's see. And so what they found there was that of our 148 North American species, 74% produce anthocyanin, Asian 38%, and European 20%. Of course, this is the botanical garden, and maybe we plant the ornamentals, with red, the maples with red leaves. This is not representative of what happens in the real biomes. But with this great paper by Paul Fine and Rick Ree, we can put that in context. We can put the percentages in the context of the species richness of these biomes. And what you see here, in this paper, which they have also Australia and so on, you have the number of tree species in Eastern North America, Western North America, Europe or Eurasia. It's Europe here, 124, measly, and Asia. And now we can put the data in context. We enlarge the data. Eastern North America has 30% of its species that have red fall color. Temperate Europe, 19%, and East Asia, 21%. And in this first paper, we didn't have the coding right, yellow and red and orange, The students documented it with photos, but we didn't do a chemical test, which is very easy to distinguish anthocyanide from carotenoid colors. Anyway, we improved that situation for the follow-up study, where we put the data in a phylogenetic context. So here are 1,500 whatever species coded for anthocyanin absence presence in a phylogenetic tree. And here is a hierarchical Bayesian model where we're treating genus and family membership as random effect to properly analyze whether geography influences whether you are red or not in the fall. And it is very clear that Europe and America, this tree species, trees and shrubs in Europe and America are highly statistically significantly different in whether they are red or not. And the same is true of Europe versus Asia. All right? And the table has all the species and their codings, and I'm very happy to send it to whoever is interested to improve, enlarge, and so on. All right. So when it comes to red leaves, geography is more important than phylogeny. But why? Why is Eastern North America so red? It's not political. All right, and so this is I never forget this moment because I suddenly had a eureka moment, and this was in St. Louis in the kitchen, and Bob and my husband and I were thinking about getting solar panels, and I said, oh no, we really have to look at whether it's worthwhile. And you can get to these websites, any number of sites where you can put in your location, and it will tell you to make you buy solar panels. Are solar panels worth it? What you should know about going solar is exactly what I looked up, and then you give in your location, and it tells you the average daily solar radiation in September for Boston or St. Louis or London or Rome. And I did that. Of course, I did Munich and St. Louis, but it's not so interesting. Even better is Boston and Rome. You are at the latitude of Rome, and you see it's highly statistically different. What Boston receives in September and what Rome receives in September is different. Now let's just to understand. So here is September radiation in watt per square meter, all right? And we will just look at an easy part here. So we have here Vermont or something here, okay? And it's kind of pale yellow. And we move over here, we come to Great Britain, Hamburg. I grew up in Hamburg, it's here, okay? It's definitely bluish, and the same move over here, we're still in the pale yellow. And here's a proper statistic analysis of this radiation per square meters in September. North American deciduous forests receive this much, and Europe this much. Temperate shrubland in North America and in Europe. We don't have that much temperate shrubland. It's clearly different. And so this gave us the idea, or please supports the idea that maybe the high percentage of North American trees that produce anthocyanins in the late fall may have to do with this wonderful radiation you receive. And I found this paper only like a couple months ago. I wished I had seen this earlier. I have to read this to you. This has all been understood since 1881. Thomas Meehan was a British gardener who moved to Philadelphia, and he gave a talk at the Academy of Sciences, and the talk was summarized in this paragraph. Okay, and the talk is about his interest in the salt marshes. Here's an American salt marsh, and it won't surprise you, it's red. Here's a British salt marsh. Same species or very closely related species of Salicornia. I have to read this. At any rate, we have in these salt marsh plants, the evidence that the plants of one country, in that country colorless, can be made to take the most brilliant colors when growing in ours, being the United States. He clearly felt himself an American. That these plants have one primary origin is certain, though the ancestry may have been separated by thousands of years. We know that plants introduced at once do not change at once. Heredity forbids this. We may assume, therefore, that it was only after some generations on the North American coast, under the influence perhaps of the American light, yes, that these European plants showed their American colors. We can see in these annual plants with a new generation every year the results in numerous generations, as we cannot see in the slower reproducing trees. Mr. Meehan thought that though we could not say we had yet reached an unchallengeable solution of the cause of autumn color in American foliage, considerations like these brought us nearer to the end. Okay, I wished I had seen this before. This is the end of the detour on radiation and senescence. Okay, back to experiments. And in another set of experiments, again using saplings, Quercus fagus and Lonicera, we applied this 5-degree warming in the spring, okay, in the spring, or in the autumn. Here's the autumn. And so what you see is the effect size in days. If you warm the quercus in the spring, you get a strong effect size on leaf out. Of course you expect this, we all know this. It's warmer, they leaf out earlier. Strong effect size. Okay? If you warm in the fall, small effect size, not surprising because we are talking leaf out here. But here's senescence. This is more interesting. So we have applied warming in the spring. Senescence is earlier. We know this by now. Okay? It's species specific. In some species you get a big effect. In some species you get a smaller effect, but it is consistent. You give them warmth in the spring, they shed their leaves earlier because they have filled their sink. This is a loose way of talking. Okay, and in senescence here, the autumn warming, if you give them warming in the autumn, senesce later, as we would have expected from your review from those 6 studies. So warmth in the autumn is still a good thing, but warmth in the spring is still much more powerful. All right? And if you give the entire warming, it has an inconsistent effect. In some species, it goes later senescence, and in some earlier senescence, species specific. Okay, but how does that translate into growth? So we killed these things and cut them up and weighed everything, including the roots, really carefully, easy, because they were just saplings. All right? So how does this experimental warming translate into growth? So experimental warming in the spring has a positive effect on growth as assessed by weighing the plants, especially the root biomass. Experimental warming in the fall had a negative effect on growth in Quercus and no effects in Fagus and Lonicera. It should be repeated with other species. It's just super interesting. So really what happens is the growth in spring. All right. On which day do you think trees switch their reaction to experimental warming? I said, spring and fall. On which day do they say, okay, now this incoming CO2 and warming, and when does the behavior switch? What do you think? What do you think?
Said June 21st.
Thank you. You saw the title of my paper. Okay, here you see. Okay, so here are the observations. Thank you. Here are the observations on 12,700 some trees of the 4 species at these sites in Europe in these years. And these sites all have daily air temperature, precipitation, soil moisture, and radiation data. All geographic. So really know what happened to that tree in that particular year, in how much radiation, how much moisture, and how much temperature did it receive, and how many cold days. Really incredible. Okay, and so you see here the relative advancing or delaying effects of a tree's monthly photosynthesis, on its end of season dates, which is the 50% leaf drop. So what happens earlier, so March, May, and so on, early in the year, delaying senescence. What happens after 21 June, advancing senescence. What happens after summer solstice is delaying senescence. And this explains, this insight, it's a new framework, explains so many contradictions in the literature, because people never separated, climate warming. You say, okay, everything warms. But the climate warming prior to the summer solstice has a big positive, it allows them to fill their sink and throw their leaves away earlier. And the climate warming after the summer solstice, okay, they keep the leaves a little bit longer if they have the water and other resources. Okay, we also have satellite data, but I'm just going to give the sentence here in the interest of time. So satellite data for the Northern Hemisphere forests from 2001 to 2018 show across 84% of the forested area, higher pre-solstice temperatures and photosynthesis have led to earlier senescence by, on average, 1.6 days. It's quite a bit per centigrade. Okay? It's actual facts based on these satellite recordings of the greenness index. While high post-solstice temperatures did not affect senescence onset, but reduced the speed of senescence, the progression, the greenness loss. And you get this only from satellite data, because you get 10% greenness loss, 80% greenness loss. You don't get that from people standing under a tree, but the satellite measures exactly the greenness index. So you get a different insight from the satellite data. So the progression of senescence is slowed by the warm climate. So now I want to come back to where I began. Okay, so we know we can use trees, they are the best, trees and other plants, shrubs, even the crops that we plant, to draw down this excess carbon dioxide, carbon that we have in the atmosphere. Okay, and the insights I have described are clearly, they will help to answer this question: what's the really achievable potential for increasing CO2 sequestration and storage? Storage, of course, in wooden buildings and bamboo bridges and things like that. Okay? The readily achievable potential can only be calculated with some reliability if we have an understanding of how the individual tree species behave and when they will shed their leaves and, of course, push out their leaves. And so in this paper, which I'm just going to show one map from this still unpublished paper, here you have the ground sourced above ground tree carbon at 1 square kilometer resolution from almost 1.2 million forest plots. These ground source real plots, people have measured stuff. This is from a database that Tom Croter and many other people, 193 other people, are involved in this database. Okay? So ground sourced above ground tree carbon that is actually there. And then, based on assumptions and machine learning, you can predict, based on the current conditions where you find this carbon, what could grow there, what additional tree carbon could grow on suitable land in these areas without humans under the current conditions. And you get this at a very fine resolution. And I'm showing you just this northern hemisphere because the opportunities to do something are, of course, easiest and best. It's the low-hanging fruit. If we consider the developed and rich countries such as Europe, Southeast Asia, China, of course, doing a lot with tree planting, right? In North America and Canada. And these, in our calculations, which are in the paper, gigatons of carbon could be drawn down just by making an effort of better managing the forests that are there, also planting some additional trees. And I want to have a fly in here. This tree planting idea has been done before, of course. For example, between 1933 and 1942, the US Forest Service and Civilian Conservation Corps, acting at the instructions of FDR, President Roosevelt planted 220 million trees in Kansas, Oklahoma, and Nebraska, in the American heartland, to fight the Dust Bowl. This was done. It was a great program. Okay? It's not impossible to do. And as we heard in your talk, what it would cost to plant a tree is actually was $250 to $900 or something. Okay, it's completely low-hanging fruit. And this is my last slide. So the insights from Botanical Gardens, all the speakers have been stressing this, and what I want to stress is, and many people said this now, the possibilities of doing experimental work on hundreds of species of trees, which we could not get at in a cheap, in an affordable way, in botanical gardens, are immense, and they are still underused. And this work is really important in the context of climate change. Thank you very much.
Thank you so much, Suzanne. Before